Hello children, I welcome you all once again in your biology class. I hope you have spent your holidays quite well and you've spent quality time, and now you're very well prepared for your studies once again. So today we will start with lesson number 5, that is structure and germination of seeds. Before holidays children, you studied about pollination and fertilization. So now we will study about seed. In the previous chapter, you studied that after fertilization, ovule becomes seed. A mature ovule is the seed. Alright. So, ovule develops rapidly after fertilization using the food reserves, and if we want to study seed structure in detail, then we have to divide it into 3 parts. So, one seed can be divided into 3 parts. First part is the seed coat, then embryo and food storage or stored food. Now, seed coat is the outermost covering of the seed. Seed needs protection, so for that purpose it has a protective covering. The covering has 2 layers. The outer layer is a tough protective covering known as testa, and inner layer is a thinner one, that is known as tegmen. This testa protects seed from desiccation, from bacterial and fungal infection, from insects. So it is a tough protective covering. This diagram you can see children, we will study each and every part one by one. Here you can see testa and tegmen. They have been labeled. 2 important things are there apart from this. One is a small scar that is present on the seed known as hilum. This hilum is the point of attachment of the seed with its stalk. And very close to the hilum you will find one opening known as micropyle. It's a small opening from where absorption of water takes place and exchange of gases takes place. Now we move on to embryo. Embryo is the young miniature plant along with cotyledons enclosed within the seed coat. Now inside the seed coat is the embryo, and embryo has cotyledons plus we'll study embryo axis. One by one we will take up. What is cotyledon? Cotyledon is nothing but the embryonic leaves, the seed leaves. Now, number of cotyledon may be 1 or 2, and this number of cotyledon present helps us to classify it under monocot plant or a dicot plant. So, monocotyledonous plant means seed which has only one seed leaf. Dicot plants, they have 2 cotyledons or 2 seed leaves. What are cotyledons? They are fleshy structures, they are food laden, and its main function is to provide nutrition to the developing embryo. Embryo needs nourishment, it needs nutrition. From where will it get it? From the cotyledon. Then I said we will study about embryo axis. Now children, this embryo axis is actually dividing the embryo into different regions. To it we find that the cotyledons are attached. So if you see the axis, you will find that it is differentiated into different regions. It differentiates the seed into different regions. The first part is mesocotyle, the point where the cotyledons are attached. Then epicotyle, the portion between mesocotyle and plumule. Now plumule is the part which will get elongated and give rise to the shoot system of the plant. Similarly, this seed will have a radicle which will get elongated and will give rise to the root system of the plant. So hypocotyle is the portion between the mesocotyle and the radicle. Plumule, as I told you, it grows into the shoot system, and radicle develops into root system. Children, this will be very clear if you see the generalized seed structure. This diagram is there in the book. You can see the two cotyledons, and there is a point, just like wings, the two cotyledons are attached. So that point, the place where the cotyledons are attached to the axis, we call it mesocotyle. You can very well understand that which region is marked as epi and which region is marked as hypocotyle. One more thing you can see, that is endosperm. So now we are moving towards understanding that what is endosperm and why some seeds have endosperm. The third part is food storage. The food is stored either in the cotyledon, as I told you, or in special food storage tissue called endosperm. Now there are two conditions. There are some plants which will have seeds with cotyledons for food storage, and there will be no endosperm. So we call them non-endospermic or exalbuminous seeds. So what are exalbuminous seeds? Seeds which have cotyledons for food storage. They do not have any endosperm. Examples are given: bean, gram, etc. Then we will find some plants which have seeds which have endosperm for storing food, and they are known as endospermic or albuminous seeds. Example: cotton, castor, etc. Now, on the basis of cotyledons also, I told you that plants are classified as monocots and dicots. Then on the basis of food storage, whether they will be having endosperm or not, again there is a further division. So now we get different types of seeds. What are they? There are 4 possibilities. Dicotyledonous exalbuminous seeds. So how many cotyledons will be there in such seed? 2, and they are exalbuminous, means without endosperm. Examples: bean, gram, pea, etc. Dicotyledonous albuminous seeds means they will have endosperm. Example: castor, cotton. Now children, in your book, castor and cotton are the 2 examples that are given. Apart from that, you can it's good for you to remember that there are many other examples. Your jute, then custard apple, papaya, etc. Then monocotyledonous exalbuminous seeds, example Elisma. There are very few examples under this category. Monocotyledonous albuminous seeds, example all your cereals and millets will come under this category. Examples are there: maize, wheat, etc. So children, today what we covered, first thing is, what is a seed? It's a mature ovule. If we want to understand the detailed structure of a seed, then we should understand first that this unit of reproduction, this seed, can be divided into 3 parts. The outermost covering, that is the seed coat, then embryo and stored food. Seed coat, there are 2 layers: testa, tegmen. Then we studied about embryo, that is cotyledon, 1 in number or 2 in number. Cotyledons are what? They are embryonic leaves or seed leaves. Prime function is to provide nourishment to the developing embryo. Then we studied about embryonic axis. I told you that this embryo axis actually divides the seed into different regions. Then we studied about food storage. In this we came across a new term, that is endosperm. Seeds may have cotyledons for food storage, or they might have endosperm. On the basis of that, we have categorized 4 different types of seeds and we studied their examples. Not to forget micropyle and hilum when we were studying seed coat. Alright children, so in the next turn we will study structure of bean and maize seed in detail. Till that time, I want all of you to revise till the portion that I have covered today. Thank you very much.